Perspective on oncogenic processes at the beginning of the cancer genomics. The Cancer Genome Atlas, in short TCGA, has catalyzed systematic characterization of diverse genomic alterations underlying human cancers. At this historic junction, marking the completion of genomic characterization of over 11,000 tumors from 33 cancer types, we present our current understanding of the molecular processes governing oncogenesis. We illustrate our insights into cancer through synthesis on three facets of the oncogenesis: somatic driver mutation, germline pathogenic variants, and their interaction in the tumor. The influence of the tumor genome and epigenome on transcriptome and proteome. The relationship between tumor and the microenvironment, including implications for drug targeting driver events and immunotherapy. These results will anchor future characterization of rare and common tumor types, primary and relapsed tumors, and cancers across ancestry groups, and will guide the deployment of clinical genomic sequencing. Introduction. In the early century of war on cancer prevention and treatment have progressed significantly, but many forms of the disease remain incurable. The advent of large-scale gene sequencing ushered in new possibilities. Beginning with coding regions, sequencing has sparked a revolution in cancer research. Genomic studies have identified numerous cancer driver genes and germline variants that increase disease susceptibility. We increasingly understand the molecular determinants of oncogenesis, including tumor suppressor inactivation and pathway alteration. Significant progress has been made in underlying driver mutations, assessing their druggability, disease subtyping, prognosis, and residual disease detection. Gene and protein expression are also key aspects. Studies have reported new fusions, alternatively spliced transcripts, express-based stratification, and implication of viral infections. Proteomic studies have made progress on subtyping, biomarker identification, and drug sensitivity and resistance. Advancements have also been made in immune response. Infiltrate-based subtyping, association of PD-1, PD-L1 with the prognosis, interaction between the immune reprogramming and angiogenesis, the immune catalytic activity. Each area shows enormous promise. The era of the first large genome sequence was called the end of the beginning of genomics. It seems fitting to call the conclusion of the Cancer Genome Atlas the end of the beginning of cancer genomics. TCGA has systematized what does this mean? How to pronounce this word?
Systematized.
Systematized large-scale genomic-based cancer research, with its projects and data on 11,000 tumors from 33 cancer types, have led to enormous advantages. The TCGA Pan-Cancer Atlas project had a special focus on the oncogenic processes governing cancer development and progression, with the 10 analysis working groups (AWG) presenting their findings together with synthesized findings from consensus somatic mutation callings, fusion detection, splicing events, aneuploidy image analysis, and the immune system in oncogenesis. Here we concentrate on three themes: interaction between somatic driver and germline pathogenic variants, links across genomic substrates, i.e. methylome, transcriptome, and proteome, tumor microenvironment, and the implication for targeted immune therapy. We begin each section with an overview from AWG results and follow with the additional analysis addressing questions not explored in individual AWG papers. The result of the Pan-Cancer Atlas project will provide a foundation for subsequent phases of deeper, broader, and more sophisticated work that holds great promise for personalized cancer care. Let's go to the results. First part, insights into germline and somatic alterations. Previous TCGA studies often concentrated on focal copy number variation rather than chromosome-level aneuploidy. The Pan-Cancer Atlas aneuploidy AWG systematically quantified aneuploidy. Let's see what is the aneuploidy. Aneuploidy is the presence of an abnormal number of chromosomes in a cell. For example, a human cell has 45 or 47 chromosomes instead of the unusual 46. Some cancer cells also have abnormal number of chromosomes. Correlated its degree with genomic features such as the TP53 status, mutational load, and level of lymphocytic infiltrate. What does this mean? Chronic lymphocytic leukemia is the most common leukemia in adults. It's the type of cancer that starts in cells that become certain white blood cells called lymphocytes. Okay, I think this in Chinese is 白血包, 白血病. And provided experimental evidence confirming some directions. Gene fusion, which can drive overexpression or create fusion proteins, are another important class of drivers. The fusion AWG systematically characterized fusions, finding that they are recurrent and disease-defining in some neoplasms. For example, SS18, SSX1, and SXX2 fusion in synovial sarcoma.
Neoplasm.
Neoplasm. Neoplasm means a new and abnormal growth of tissue in some part of the body, especially as a characteristic of cancer.
Neoplasm. synovial,
Synovial means relating to or devoting a type of joint which is surrounded by a thick flexible membrane, forming a sac into which is secreted a viscous fluid that lubricates the joint. Uh, synovial, synovial, synovial,
synovial,
synovial, synovial, synovial, synovial, synovial sarcoma, synovial sarcoma, right? Let's say how to pronounce sarcoma.
sarcoma,
Sarcoma, a malignant tumor of connective and other non-epithelial tissues. The type of soft tissue sarcoma is a rare cancer. Only about 1 to 3 individuals in a million people are diagnosed with this disease each year. It can occur at any age, but it is more common among teenagers and young adults. Synovial sarcoma, synovial sarcoma, synovial sarcoma, synovial sarcoma, synovial
sarcoma,
sarcoma. In others, fusion drivers are present in small subsets of the tumors. ALK or ROS1 fusion in lung adenocarcinoma.
adenocarcinoma,
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 adenocarcinoma, adenocarcinoma. The accompanying mutation events and how they differ among cancers provide functional insights. Two other AWG systematically characterized germline and somatic variants across 33 cancer types. They generated and analyzed 1.5 billion germlines and 3.6 million somatic cores, making TCGA Pan-Cancer Atlas the largest resource for investigating joint variant contribution to the cancer. The germline group highlighted the two-hit hypothesis through loss of heterozygosity (LOH) and compound heterozygosity, rare copy number events, and additional evidence supporting variant pathogenicity. The somatic data set anchored a comprehensive dataset using 26 bioinformatic tools, identifying 299 driver genes and over 3,400 oncogenic mutations. Similarly, the Pan-Cancer Atlas germline group identified more than 800 pathogenic and likely pathogenic germline variants in 99 predisposition genes affecting 8% of all cases. Property of oncogenic germline and somatic variants. Here we use the 299 driver and 99 predisposition genes to study the interaction of germline and somatic events in 9,389 samples. Many predisposition genes play roles in genome integrity and alteration in these genes represent a higher fraction of germline variants versus somatic drivers. Highlighting the role of genome integrity in cancer predisposition. What is cancer disposition? The liability or tendency to suffer from a particular condition, holding a particular attitude or acting in a particular way.
Predisposition. Predisposition.
Predisposition. The remaining somatic alterations are largely from genes involved in cell cycles, epigenetic modifier, metabolism, oncogenic signaling, and transcriptional-translational regulation. We surveyed the frequency of cases showing disruption of genome integrity in individual cancer types. One of the 8 molecular process categories examined, genome integrity dominates both germline and somatic alteration in ovarian serous cystadenocarcinoma. Okay, I don't know how to spell this. This is too complicated. Ovarian serous cystad.
Ovarian serous cystadenoma.
Okay, I don't think this is the correct way to pronounce this. Let's see whether there's a YouTube about this one.
In contrast to the previous case that we saw, in which the tumor was at least partly cystic, but on cut section and microscopy was terribly malignant, here's another tumor of the ovary that looks very cystic. In fact, the whole thing looks like a cyst. So let's take some sections through the wall of the cyst and open it up and look at it under the microscope. And if you had cut through the wall of this cyst, which this is here or here, you might have noticed that the internal surface, in contrast to being nice and smooth and shiny, as the external surface was, might have had these little fuzzy granular areas called frequently excrescences, which look like they're little fuzzy thickenings or nodules on the inside of the cystic surface. Notice that once again it's papillary. Notice once again that the cells do not make mucin. They appeared to be nice and blue. If they were making mucin, the cells themselves, including their cytoplasm, would have a whitish consistency. These are bluish, so these are serous cells rather than mucinous cells. But also notice, in contrast to our other one, which was papillary serous cystadenocarcinoma.
Oh, let's say just say this OV. This is the OV due to BRCA2, BRCA1, BRCA2 predisposition variant and a high fraction of TP53 mutation, other cancers are further skewed with respect to percent of cases carrying mutations involving genome integrity. 4% of samples in lung squamous cell carcinoma have germline compared to 80% non-somatic. Gene damage response pathways. Most predisposition genes affecting genome integrity, 60%, belong to the core DDR, gene damage response genes. Several show high germline variant counts, including BRCA2, BRCA1, CHEK2, ATM, BRIP1, PALB2, and PMS2. When considering germline and somatic mutation jointly, the most frequently mutated genes are BRCA1 and BRCA2. Together have 571 samples, somatic, and 153 germline mutations. We grouped samples with the germline mutation, somatic, and non-low impact mutation in these two genes by cancer type and established association between age of onset and somatic mutation load. Patients with the germline BRCA1-2 mutation develop cancer at a younger age compared to wild-type samples in OV, LU, SC, and BRCA false discovery rate, respectively. Mean age of diagnosis in patients with germline mutation is 500. It's 54.4 minus and plus and minus 13 years standard deviation, compared to 62.3 plus minus 13.4 years when the mutation is somatic across the pan-cancer cohorts. As expected, germline or somatic variants associated with higher mutation load across cancer types being observed in OV samples with germline BRCA1-2 mutations and BRCA STAD somatic.